Alright, this is the first lecture I'm going to do, and this is on the very basics of enzymes, and really I'm just going to try to skim the surface. I don't want to get too involved here. So a lot of this should be review. A lot of times we think of enzymes as just being, a protein, but that's not always the case. Sometimes they're also nucleic acids, so it's important to note that enzymes could be, either or. In the picture here, I have a picture of pyruvate kinase, an enzyme you might be familiar with from biochemistry. If you haven't heard of it, it's totally fine. But essentially, pyruvate kinase is a critical step in the formation of pyruvate, and we will be seeing it again later in this lecture at some point. Some basic features of enzymes. They decrease the activation energy of a reaction, and they increase the rate. And so if we look at this reaction coordinate, we have on the x-axis, we have the progress of the reaction. On the y-coordinate, we have the free energy, represented as G. And we have the reactants we can see are starting are in higher energy than our products are. And therefore, in biology, this is sometimes referred to as an exergonic reaction. So we're giving off some form of energy. And the change in energy from the reactants to the products is this delta G. That's the thermodynamic change in free energy. I will repeat that. That is the thermodynamic change in free energy. Okay, so it's from the reactants to the products, that's the change in the free energy. And we'll see in the next slide that the enzymes do not affect that. Enzymes do not affect this. This is going to happen either way. So you can see this blue curve is the enzyme, and the pink curve is without the enzyme. We can see the activation energy is increased, right, for the reaction without the enzyme, whereas the reaction with the enzyme, the activation energy is lower. But in both cases, the total delta G is the same. So that if you're kind of confused by that, I suggest maybe watching that again, and maybe you can even do some research online, but this is something that you kind of should have a feel for it at this point. So here we see that the process by which these enzymes interact with substrate is kind of unique. So you have this enzyme substrate binding that occurs, and essentially this is like an equilibrium process. So substrates will bind, but then they might get kicked off. They might go back this way, or they might go forward. And if they go forward, sometimes the substrates reconform due to the binding energy that occurs between these two, and then they get split off, and you get products released essentially. And this is a very generic scheme, and we'll talk about a lot of specific examples of this. You can see that enzymes do not alter the equilibrium of a reaction. This goes back to the last couple slides. They'll change the rate, but they won't actually change the equilibrium, and that's because the equilibrium is based on these concentrations. And essentially, there is a set concentration. There's an equilibrium constant for a reaction, and we can change the rate that we get to that equilibrium, but we can't actually change the equilibrium itself through the enzyme. And so we can see that there's sometimes other partners in crime for these enzymes that help them function, and those are called cofactors. And there's really two types. You have metals, and usually those are ions, like magnesium, for example, is a very common one, again going back to the glycolytic pathways. But sometimes we have coenzymes. You have different vitamin derivatives that you might see, and other molecules you'll see at some point. And really, the coenzymes encapsulate both the vitamin derivatives and the smaller molecules. And here, it's a little messy, but I just kind of drew some things together, and I'm a lefty, and I kind of have the doctor's handwriting, so to speak. But I think you kind of get the idea. This is similar to the graph we saw before, and the red curve, as you can guess, is the reaction without an enzyme, and the green dotted line is a reaction with the enzyme, and they're going and ending at the same place. A as the reactants, B is the products. They're ending at the same energy, right? The brown delta G you see there is the thermodynamic free energy, and then there's the the activation energy, which is sometimes referred to as delta G, and then they have that transition symbol, which is this symbol here. It's used on these delta G's for the catalyzed and uncatalyzed reaction. And that delta G, the free energy of the transition state, which is the highest point in the reaction, is going to change, and the enzyme will really control that. It'll bring that delta G transition state down, as you can see here. All right, let's try a follow-up question to see how well we understood some of the material. I'm not going to read the whole question out loud, but what I would suggest is potentially pausing the video here and seeing if you can reason through this and find an answer that you like. And I'll go over the answer, but before I do, I'll count to 5, so I'll give you a chance to pause, read through, and see if you can come up with something. 5, 4, 3, 2, 1. So the answer to this is A, increased rate of pyruvate production. And I guess the key really here is not to get bogged down in some of the terminology. So we talked about pyruvate kinase in the first slide. And I said you'd see it again. And here it is. And really, what you have to understand is pyruvate kinase, like I said in the first slide, and you can kind of decipher here, it's an enzyme, okay? And the enzyme is being activated by this guy here, this 1,6-phosphofructobisphosphate, which is not really important, okay? He's just activating the enzyme, all right? And then we can see that that reaction, the reaction is this PEP forming pyruvate. So pyruvate kinase is the enzyme that allows for PEP to turn into pyruvate. Essentially, that's what we see here. Now we're trying to find out which of the following options would be affected when this pyruvate kinase is activated. Okay, so when the enzyme is activated, what's going to be affected? So the first option, increased rate of pyruvate production. Does that make sense? Well, if I activate the enzyme, I'm going to get a greater rate, right? We remember that from the first slide. We're going to increase the rate of the reaction. So I'm going to have an increased rate of the product forming. Obviously, B is the opposite, so that's not true. C, increased activation energy for the conversion of PEP to pyruvate, right? It's not true, right? I'm decreasing the activation energy when I use an enzyme, so that can't be accurate. D, increased rate of PEP production. This is tricky. You have to understand that in the question it says, if you're not familiar with this enzyme, that is, in the question it says to allow for further production of pyruvate from PEP. So PEP is the reactant, pyruvate is the product. So if I'm activating the enzyme that allows this reaction to continue, I am going to get a decreased concentration of PEP. E, increased rate of the 1,6-phosphofructobisphosphate production. That 1,6-phosphofructobisphosphate has nothing to do with the, or at least the production of it, is completely irrelevant to this reaction. All that player does in this reaction, that component does in this reaction, is really to activate the pyruvate kinase. Okay, so it's not actually a key player that does something in the reaction other than just activating the enzyme itself. Hopefully that helped. The next video that we will do, we'll talk a little bit about the reaction velocity for enzymes.